The overall goal of this procedure is to detect foodborne pathogens in large volumes of agricultural water. To do this, a modified Moore swab, referred to hereafter as an MMS, is assembled by placing a folded piece of gauze or cheesecloth inside an MMS cassette. Next, at the sampling site, target bacteria are concentrated by passing large volumes of water through the MMS using a peristaltic pump. The ends of the MMS are then tightly capped and transported back to the lab. Selective or non-selective enrichment broth is added to the MMS, which is incubated for 18 to 24 hours. The enriched cultures are then collected, and for E. coli detection, lysates are prepared. These are added to micropad microspots impregnated with substrates for colorimetric enzymatic detection and incubated for up to 3 hours. The micropads are then inspected and scanned. Ultimately, visual and quantitative analysis of the microspots reveal the presence of E. coli, Salmonella species, and L. monocytogenes at levels as low as 0.1 CFU per milliliter.
The main advantage of this technique over membrane filtration and culture on solid media is that larger volumes of water are sampled. This increases the sensitivity of the assay and also decreases the enrichment time. The time to detection is also decreased to within 24 hours, as compared to 24 to 48 hours for the membrane filtration method.
In addition, the micropads can be used to simultaneously detect 3 different foodborne pathogens, which cannot be done on the traditional solid bacterial medium.
The implications of this technique extend toward analysis.